In our last video, we discussed why it is important to better understand the biology of glioblastoma. One important point we made is that glioblastoma and many other brain cancers contain cancer stem cells that drive the growth of tumors. So the question is, how do we target these cancer stem cells? Two examples of how scientists are trying to find new and effective drugs for brain cancer is by 1. Using drugs to target the unique functions of cancer stem cells, and 2. By repurposing existing drugs. Let's explore these concepts further. We have learned that normal stem cells had unique signals that give them the power to continuously multiply and grow. We now know that cancer stem cells have the same power which ultimately drives their tumor growth. Using this knowledge, we can use drugs that will block specifically stem cell signals in brain cancer. The result? We find that this type of treatment forces cancer stem cells to turn into mature cells that can no longer divide or grow. So instead of trying to kill all cells, we are using drugs that simply coax cancer stem cells to turn back into normal cells. Another way to find better therapies for glioblastoma is by repurposing existing drugs. When we repurpose drugs, we test drugs that have already been used to treat other diseases. This is based on the notion that drugs designed for other diseases can also work on the same signals in cancer cells that have gone awry. This is especially important because it can take up to 16 years to develop a new drug and make sure it is safe and effective. So if we can use an already FDA-approved drug and prove it can work on cancer cells, this could bring new treatments to patients faster. So how do we find new therapies for brain cancer in the lab? From the tumor, we can take out the cancer stem cells and grow them on a dish and apply different drugs and ask questions like, do the cells stop growing? Are they dying? How and why is the drug working on these cells? Let's look at an example. One drug used to treat Parkinson's disease was developed to target a receptor known as the dopamine receptor D4, a normal part of a cell communication pathway in the brain. When this drug was tested on glioblastoma stem cells, scientists found something interesting. Glioblastoma stem cells no longer grew and divided. In fact, the dopamine receptor was found to be needed for cancer stem cells to survive. And if we remove it, then these cells can no longer work properly, they lose their stem cell traits, and eventually begin to die. So what's next? Studies like these will help the research community better understand the importance of cancer stem cells. By identifying new targets in glioblastoma, this will spark more studies by other scientists, and together these preclinical studies will inform new clinical trials for testing of new therapies in glioblastoma. Thank you for watching, and we hope you learned a little bit about brain cancer biology and how we are using the knowledge of cancer stem cells to discover more effective therapies for this disease.